The next speaker is a bit of a modeler as well. So the next speaker is Lydia Turley, who is a PhD student here at the University of Otago, and not super far into it, but this is a kind of, she's going to give us like an introductory talk and some of the simulations that she's been doing.
Cool, thank you. So I'm a PhD student at University of Otago, working with David Orlovich and also David Bryant, Matt Lacombe and Janice Lord. And I'm looking at mathematical models for populations and genetics and fungi. So I wanted to introduce a bit about what these models are doing, where they're coming from, then talk a bit about my, my fungal specific models and share some preliminary results there. So population genetics, as the name suggests, is all about studying properties of populations using genetic data. So we can look at the distribution and frequencies of various genes and use that to draw conclusions about how the population is evolving and interacting with other populations. And all of this is based on models of gene flow and population dynamics. So these underlying models are really important in how we link the what we observe to the conclusions that we draw. So if we want to do a population genetic study, we'd start by getting some data. So we'd go out and sample a bunch of individuals from our population and get some kind of genetic data, typically DNA sequences, which can, stuff which has some variance within this population that we're looking at. And then we go and calculate some statistics. So the kind of things we can look at are the frequencies of genes, the heterozygosities, the probability that if we pick two different copies of an allele of, two different copies of a gene, are they going to be the same? And we can look at differentiation between different populations. And from these statistics, we can draw conclusions about what we're actually interested in as biologists. So these might be questions about things like colonization history. So if we've got something that's in a species that's invading in somewhere, you can look at where it's come from and how it's moving using this. We might look at things like population subdivision. So at what geographic scale do we have barriers to gene flow? And the models that I mentioned are what connect these statistics to the conclusion. So they're telling us how we're supposed to interpret this, or how we're able to interpret the statistics that we get out of there. So the standard model in population genetics is the Rat-Fisher model, which may be familiar in the guise of Heidi-Weinberg equilibrium. And this is built on some assumptions like all models. And this particular one says that we've got discrete non-overlapping generations, we've got random mating, and it's given in terms of haploid individuals, but it works fine for diploids. And it also assumes that there's nothing exciting going on in there. And so if we've got some hermaphrodite annual plant in a very isolated population, then that's probably quite a good model. But if we've got other things that are kind of behaving quite differently, it might not be such a good model. So models are always an approximation of reality. It's never going to be perfect, but we do need it to be good enough. That's all we want, something that's good enough. Issue is when we're dealing with fungi or with various other organisms, sometimes these assumptions can be quite badly violated. So we know that our fungi, some of our fungi, particularly mycorrhizal ones, can be really long-lived. The mating can be really weird and not random. We have different issues with ploidy and things. And if these assumptions here lead into the statistics that we calculate and how we're supposed to interpret that. So if our model says that one thing's going on over here and our actual population that we're studying is over here doing something completely different, then that might make these statistics and the way we're interpreting that meaningless, because we're saying that this statistic that we calculate from this population says one thing, but actually the population's doing something completely different. So I'm particularly interested here in fungi, we all are, and we know that fungi are behaving quite differently. We want to know if this is enough to be a problem for these traditional Rat-Fisher-based models. So what I've done is developed a new fungal model for population genetics here. We've got a really simple Basidium mycete life cycle. I started with something nice and easy. We've got a little sporocarp. It produces spores. These are haploid. They can then germinate and form a monokaryon. So this has got one haploid nucleus per compartment. These can meet and mate and form a dikaryon. So this has got two distinct nuclei in each compartment. This is the long-lived, persistent stage of most of the Basidium mycetes. And then the dicaryons can form mushrooms. And so in here, the nuclei fuse and undergo meiosis. That very simple basic kind of approximation of what's going on in reality as the basis of all models. What I want to do is turn this into a model. So I'm assuming that we've got separate monocaryon and dicaryon populations. I'm assuming that we've got fruiting is happening seasonally. It kind of is. It makes life easier. And I'm allowing for a really long-lived persistent individuals. So each season we have some number of fruiting bodies which are produced, and their genotypes are drawn from the underlying dicaryon population. So we're just having some dicaryons produce mushrooms. And then going from one season to the next, the monocaryon population we update as a mixture of the previous year's monocaryons and things that had come from the spores. The dicaryons are then a mixture of the previous year's dicaryons and things that have been monocaryons and some things that have been spores. So each of these gets updated. Next step is to turn this, this model description of what's going on into mathematics. So here we've written some equations that are just describing exactly what I said, but in mathematical terms. So the y's and the x's, we're describing the proportions of whatever allele we're looking at in the population as a random variable, and we can see how that changes from time to time. We can take this set of equations and express parts of that using matrix algebra. So we get some matrices like these. And then we can look at what happens to some of these statistics that we might look at. So we can look at the expectation, the variance, and over here I've got an equation for a form of the heterozygosity. And so we can see how these change from through time. And from that we can look at what's happening to our statistics and how is that relating to what's actually going on in the population. Next thing I can do is code up a simulation. And so I can simulate what's going on in here, and it just makes it a bit easier to go on. So here I'm producing 49 fruiting bodies, and these all have two copies of the genome. So we've got 98 alleles. And we started around here. And so we had some proportion of the allele in the underlying dicaryon population, about this initially. And then we draw some fruiting bodies randomly. These update, and after several seasons I've created 1,000 copies of what's going on. So I'm running the evolution 1,000 times, and so after the first couple of things we've got a bit of variation in the distribution of allele frequencies in our fruit bodies, and through time this kind of spreads out. We start seeing that some get stuck, the allele dies out, and that increases through time, and then eventually some of them start getting fixed. So this goes going on. These two parameters here, the alpha and the S, are the amount of our dicaryon and monocaryon populations which get replaced each season, so the amount of turnover. So in this case, they're completely replaced every year, and it's just our fruiting bodies going on. This particular parameter regime is exactly identical. Our fungal model is exactly identical to the rat fisher model that we described. So what I can do now is look at what happens to some of these interesting statistics, and the heterozygosity is the obvious one to look at. So it's kind of decaying over time, and I've plotted the simulations of the fungal model in blue. I've also plotted the rat fisher model in red, and the expectations under the rat fisher model in yellow. And we can see that they're all agreeing, which is perfect. That's what we want, because they all should be exactly the same thing. Or different simulations of exactly the same thing. What I can do is change these parameters. So as soon as our alpha and our S decrease, this means that there is some persistence in our monocaryon and dicaryon populations. There are some individuals that last from one season to the next and don't get replaced. And this is effectively just kind of slowing down the change each year, which is what we observe here. So we can see that all these little normal distribution-y kind of things take a bit longer to spread out and get fixed. Back. Here we go. So then we can again plot the heterozygosity, and because I changed these parameters, we're no longer looking at exactly identical to the rat fisher model. So what we can do is try and calculate some equivalent population size such that the rat fisher model behaves the same way as our fungal model. And over here, I've got an approximation of what I kind of think this number should be, but it's, as we can see, it's looking pretty good, but it's not quite right. So this is kind of going to need a bit more work going on, because if we can find some population size such that the rat fisher model behaves exactly the same as the fungal model, then that's fine. We don't need the fungal model. We're okay using the rat fisher model. And that's good to know because that will be the underlying model that's gone into all the previous work in this kind of field in fungi. And so if it's working fine, that's actually awesome. But if we just can't find anything and there's always going to be this bit of difference, then we do need special models for fungi. So we've got a little bit more work. I've got a little bit more work to figure out exactly if we can get these matching perfectly or if there is an intrinsic difference going on. So we're onto fungi here, actual fungi. The next step I want to do is look at what's going on in real fungal populations. So I need to go out and get some genetic data which allows us to differentiate individuals of the same species. What I'd love to do is dig out the dirt and get all this mycelium kind of growing around in the soil. Unfortunately, this is not so easy to do because I need to first identify which species it is in order to then be able to get out the genetic data which will differentiate individuals. So I'm kind of restricted at the moment to sampling fruit bodies because at least we can know a priori what species I'm looking at. The issue there is that this is potentially quite a biased sample of below ground diversity because we don't know if all individuals are producing fruiting bodies at the same rate or like what kind of relationship there is between fruit body production, mushroom production, and the dikaryotic mycelium in the ground. And so my model needs to be a bit more robust to this kind of subsampling method. I found a model organism. I want to look at Amanita muscaria. This is abundant. It's got easily identified sporocarps. Even I can figure out what they are. It's ectomycorrhizal, associated with pine, exotic, and is currently been observed invading into native beech forest. So this means that there are some really interesting potential questions out there in terms of, are the invasive populations behaving the same or differently to stable populations in pine forest? We could look at where has it come from, how is it behaving, all those kind of things. It's been genotyped, which makes it really easy for, is very helpful for getting the genetic data that I want. And I have recently discovered that it's a whole lot less abundant when I'm actually hunting for it than when you just see it popping up everywhere. I'm sure all of you know that though. So my plan is to go out and sample a bunch of these sporocarps from around the Dunedin area. Got to extract the DNA and sequence them using genotyping by sequencing. And then I can look at the population genetics. So we can look at things like the size distribution of the individuals, see if we can actually tell that from the mushrooms. We can try and estimate the model parameters, so kind of see how much turnover in the dicarion population there is each season. And it would be really cool if we can do things like predict the distribution of fruiting bodies based on the previous year's data. I'm not sure if that will be possible, but that would be awesome. I think that's about all I had to share today. So questions or suggestions?
Great, thank you, Lydia. That's fantastic. All right, I have actually got a question. I know I'm supposed to, I'm your supervisor, but I've got to say, every time we have a meeting, Lydia absolutely drills into my, what is largely a textbook knowledge of what the fungi actually do. And I think most of what we've found is only some of them do what they say in the textbook, right? So like, I'm just trying to think with your simulations, then as an example of the application of this in the future, like let's say climate change causes mushrooms to fruit at a different time or to fruit less, for example, then that's the sort of, then the effect of that less fruiting on the population as a whole is the kind of, you could predict that with your model, couldn't you? Is that the idea?
Yeah, we could definitely look at, what happens if you change the number of fruiting bodies that it produces, or the relationship between the fruiting bodies and the underlying mycelium, and you can look at how that changed, we could treat that as a function of time, allowing that to change.
Yep. Okay, great. Okay, Kath has a question in the chat there. In terms of finding Amanita muscaria, would it be useful to put observations from Dunedin on iNaturalist, or are you looking for specific habitats, i.e. pine forest?
I'm kind of focusing on a couple of specific populations so that we can deal with specific, nice, well-defined populations for the population issues. And also I need fairly good quality genetic data. So things like observations on iNaturalist. It will tell me that there is a mushroom there, and that it is an amanita, but I won't know how it's related to any of the other individuals in the population. So it would be cool, awesome, but probably not helpful for what I need.
Great. And David Harris said, great talk, Lydia. Have you considered sampling soil for Amanita mycelium near pines as well?
I would really, really, really love to. That sounds like exactly the data I want to have. However, the issue is that I need to be able to differentiate between individuals of Amanita. So if I need to be able to extract the DNA and then know that I'm looking at Amanita in order to get the sequence little bits. And if I just kind of dig up soil and get some kind of mycorrhizas or stuff and don't know what it is a priori, I'm told that we can kind of genotype it to the level of identifying which species are out there. But I'm told that it's not really possible to get the level of individual differentiation stuff while also knowing that I'm looking at the right species.
Yeah. So one thing that we could do, I guess, is to plate out on agar and separate individual colonies of all the fungi that are there, because Amanita muscaria does grow. And then once you have isolates from the soil, then you can do that too. That'd be make you into a real lab monkey instead of a modeler, wouldn't it? That's awesome.
Yeah, okay, I guess I could do that.
Yeah, yeah. Okay, Ian Dickey has a question. Can a single monokaryon mate, oh God, where's it gone? And here we are, mate with other monokaryons? Yeah, doesn't it? And does that affect the model? Can a single I think that's what happens, isn't it? Can a single monokaryon mate with multiple multiple other monokaryons? Yes, that's right, isn't it?
Yes. So this model is extremely simplistic there, and it's definitely the mating process are way more complicated than what's going on. However, how do I move my things? When I have this model here of what's going on, basically I'm just looking at each copy, like each nucleus. It can move from being in a monokaryon to being in a dikaryon, and I'm not actually interested at this point in how it moves, whether it moves via mating with another monokaryon or via mating with a dikaryon or whether mating with whatever else. Once I adapt the model a bit, improve it, and deal with kind of linkage of things, then we will start to care about if one nucleus is in a dikaryon, then it's attached with something else and it's associated with some other nucleus. And then I might start to care about those kind of things. But at the moment, because my model is so simplistic, it doesn't matter. But that's definitely a major issue that's going to be very complicating when I want to consider that and improve things.
Yep, it was Lydia's PhD that made me learn about this Buller phenomenon where the dikaryons can escape and one monokaryon can grow out of a dikaryon and mate with another. Oh God, yeah, it's complicated.
Yeah, and it doesn't even seem to be very consistent as what it does there.
Right, yep. And then you have diploid mycelia too in some species. Okay, following Ian's question, what about diamond mating? Right, yeah, well, that's what we just discussed there, that's right. And Ian's curious if a single spirocup can be two dikaryons at once. Yeah, no, that's an interesting question, and you've got to go, yeah, because the ascomycete, the lichens can do that. The thallus can be multiple individuals growing together there. Awesome.
Well, if we find any, then that'd be cool to know.
It would. Fantastic. All right. Yep. All right. So I think that is, we better leave it there.